Her research has resulted in the development of an immunotherapy that's currently in preclinical testing. Furthermore, her team has developed a bioinformatic tools for prioritizing drugs that could be repurposed for the treatment of head and neck cancer, and she has received research grants from both the MRC and the UK as well. And she also received awards including the Norman Romeo Education Trust and the L'Oreal for Women in Science Award. She is also a fellow of the International Academy of the Oral Oncology and the Academy of the Science Malaysia. She is currently the co-chair of the World Academic Science and the Academy of the Malaysia. She is currently also an honor member of the Young Scientist Network in the Academy of Science Malaysia. So she's going to share with us today cancer genomics and recent advances in screening, diagnosis, and therapy. Welcome, Professor Chong.
Thank you very much, Professor Choi, and I would like to thank Professor Thong and the organizing committee for the kind invitation to come and present some of our work at this conference. This is not the typical conference that I attend, so therefore I think my role today here is to talk a little bit about what goes on behind the scenes, the research that occurs before a test is actually put on a panel. So what sort of research goes on, and how are genetics or genomics underlying diseases identified. So today in my talk, I'm going to talk a little bit about what is the status of cancer genomes in the world, and what are some of the clinical implications coming up from all this work on cancer genomes, and why I think we really urgently need to invest in Asian genomes, or trying to understand the genomes or the mechanisms underlying cancer as it happens in our population. I will give you 3 very short briefs about our projects, how we take Asian breast cancer genomes to immunotherapy, and how we identify unique cancer antigens to develop cancer vaccines for head and neck cancers and other cancers, and also how some of these genetics studies have resulted in new information that we really seriously need to think about when we are looking at our clinical practice guidelines. So when we talk about sequencing, we typically talk about sequencing in 2 areas. Either the germline sequencing, where typically this enables us to identify high-risk individuals. A lot of this has been talked about in the conference today. And we also have tumor sequencing, or what we call the cancer genomes. So tumor sequencing is what I'm going to focus on in the first and majority part of my talk. And tumor sequencing is the way to try and understand what is happening within the cancer cell, so that this can help us enable selection of precision and targeted therapies. For example, targeted therapies towards certain gene fusions or amplifications. So one thing that I would like the audience to keep in mind is that cancer is a very heterogeneous disease. So even if we talk about breast cancer or head and neck cancer or lung cancer, no 2 cancers are the same. And in fact, we need to categorize some of these cancers into sub-classification so that we are able to give the cancer patient the right treatment at the very start. And of course, cancer can be characterized by either mutations, gene deletion, gene amplification, or also epigenetic regulation. And we have not short of cancer studies that characterizes the genome. I'm going to talk about a few studies here, and the first one is the International Cancer Genome Consortium, which then developed further into the pan-cancer whole genome sequencing. And you can see from the map that many countries are participating in this kind of consortiums, and the majority of these sequencing anatomical sites depend on what are the key cancers or the cancers that are important for each of these countries. So for example, the area that I work in is head and neck cancer, and specifically oral cancer. And oral cancer is the #1 cancer in India. It's the national cancer. And you can see that India has taken the responsibility to sequence oral cancer. And of course, the Western world has a lot more resources, and you can see, for example, the United States of America has taken on many, many more cancers than the areas that, in the regions that we live in. So currently, the pan-cancer genome has more than 2,600 genomes sequenced across 38 tumor types, and they have identified about 700 mutations that co-occur in different cancers. And importantly, some of these mutations are outside coding regions and will not be detected if you use targeted sequencing. And they have concluded that each cancer could have about 4 to 5 driver mutations, and about 5% of tumors have no driver mutations. Just want you to keep that in mind as I go along. So these studies, the TCGA or AACR-Gini comes out of the United States of America, and there's a very nice portal for those of you who work in cancer called the CBio portal, and all sorts of cancers, more than 40 different kinds of cancers, you can already download and look at their genomes. And what is really nice about AACR-Gini is that there's about 170,000 patients, and these patients are unique in the sense that they also have real-world clinical data. So by looking at this portal, you can actually follow patients and query this database so that you can actually make some sort of armchair research for hypothesis generation before you do very expensive wet lab experiments. But what is important about these databases is that although they are huge, for example, AACR-Gini has 170,000 patients, less than about only 20,000 patients are non-white patients, because the majority of the population is white. Another study that has just come out in terms of their reporting in terms of cancer genomes, back to back, many papers are coming out from the UK 100,000 Genomes Project. And again, this is in excess of 30,000 cancer types. And what they found is that 55% of the cancers sequenced have at least one clinically relevant mutation. This means that perhaps cancers that we think may not have targeted therapy, or we do not know the prognosis, may now be bundled together with other cancers with similar mutations so that we will know more about how to treat these kind of cancers. And 15% of these patients would have been eligible for a currently approved therapy. So this is a good opportunity for drug repurposing. What is really important about these kinds of genome projects is the fact that maybe there are some drivers in certain kinds of cancers that you did not know those cancers are characterized by these specific drivers. For example, some cancers have the HRD or HRD gene repair signature. They may not necessarily have a BRCA1 or BRCA2 mutation, but nevertheless, they would have a DNA repair defect. So these kind of drivers that are found in cancers that were not known to have these kind of drivers now could be an opportunity for repurposing of drugs or even diagnosis. And importantly, these sorts of genomic studies also give rise to new driver mutations that were not discovered previously. And therefore, this gives us an opportunity to expand the druggable genome. Again, because this 100,000 Genomes Project was done primarily among cancers that are very commonly diagnosed in the UK, and patient ancestries and geography, as we know it, can affect mutagenic profiles of this tumor. Therefore, we may not necessarily be able to use databases such as AACR-Gini or even the 100,000 Genomes Project to extrapolate directly to our populations. For example, you can see that head and neck cancers that is more common among Asians are actually at the bottom of the list here. So now that we have a lot of cancer genomes, what can we do with this information? So cancer genomics have been used extensively for early detection. So for example, for circulating tumor DNA for diagnosis. And now even multi-cancer early detection or MCED tests are being developed and tested with a sensitivity of about 50% across 50 different kinds of cancers. This information has also been used to try and come up with AI algorithms to diagnose cancers of unknown primary, because 3 to 5% of all cancers are in this cancers of unknown primary group. And by knowing what is the primary cancer, we can quickly treat these patients. What about prognosis? So transcriptomic information such as those for MammaPrint or Oncotype DX, which will enable us to identify breast cancer patients that require adjuvant therapy, are already used in clinical practice. And cancer genomics have also been used for predictive. So for example, tumor mutational burden and also MSI and MMR cancers are now used as a tumor agnostic test to identify patients that will benefit from immunotherapy. And of course, very importantly, even after treatment, cancer patients are at risk for recurrence, and this information has also been used for monitoring patients, not only for disease recurrence, but also to identify genes that are mutated, causing resistance to current therapy. Unfortunately, as I mentioned, many of these genomic studies are not focused on the Asian population. And although Asians make up 60% of deaths due to cancer, it only makes up about 10% of research. And 70% of the deaths due to cancer are going to occur in low and middle income countries. So this is really something that we really need to look at seriously. And we know that investment in cancer research is heavily skewed towards high income countries, mostly done among Caucasians. What about clinical trials? So clinical trials are also heavily skewed. As I mentioned, Asians make up the global population, large part of the global population, but if you look at where clinical trials are done, again, Asians make up the minority. This is really important because ancestry really contributes to the prevalence of certain cancer-associated genetic alterations, and I'm going to use one particular example, which is the epidermal growth factor receptor, which is the target for cancers such as lung cancer and colorectal cancer, among others. You can see that overall mutational frequency of EGFR differs based on ancestries of the patient with cancer, where the highest is among East Asians at 50%, whereas at the lowest will be amongst the European and also amongst the African. And this frequency of this mutation curiously follows the pattern of migration. If you follow this blue dot, which basically depicts the Asians, you can see that those Asian migrant populations will have high EGFR mutations compared with those migrants that have the European ancestries. And looking at a paper comparing EGFR mutations among the Chinese in China and also in America, you can see a stark difference, 58% or close to 60% among the Chinese and only about 22%. And this is one of the genes that are very important in terms of cancer therapy. So Cancer Research Malaysia is a not-for-profit organization, Malaysian run, and our goals really is to accelerate treatment for cancers among Asians, and I'm going to give you two examples of how we have done that thus far. So the first story I'm going to tell you is about Asian breast cancer genomes to immunotherapy, and the second one would be how we have identified unique cancer antigens that are now being developed into cancer vaccines and awaiting or poised for phase 1 first-in-man clinical trials. So in 2012, we started a cohort of Malaysian breast cancer patients, And of course, breast cancer is the #1 cancer in Malaysia. And we collected about 1,000 Malaysian patients with breast cancer, and we characterized their tumor genomics and also transcriptomics by sequencing these tumors. And at the moment, what we found is 3 major findings as a very brief description of the paper. Curiously, we found a lot more HER2 positive cancers and more TP53 mutations, and this is quite curious and different from the Caucasian breast cancer patients, where PIK3CA mutation, which is the second most common cancer here, most common mutation among our cohort, but usually PIK3CA is more commonly seen among the Caucasians and TP53 less seen. And importantly, because immunotherapy is now mainstream for more than 25 different kinds of cancers, we also found our breast cancer tumors to be more immunogenic compared to, let's say, the Caucasian breast cancers. Why is this important? As we know, HER2 cancers are typically treated with Herceptin, and this is important because Herceptin is a form of targeted therapy, and therefore, knowing this, that more Asian women need HER2 targeted therapy, we have to work with our Ministry of Health to ensure that such therapies are available for our patients. TP53 mutations typically are associated, because it's a huge tumor suppressor gene, is typically associated with poorer survival. And this then now gives us some hint of potential biomarker for targeted therapies. And importantly, because we have more immunogenic tumors, Asian women may respond better to immunotherapy. And previously, when we look amongst the genomes for breast cancer patients, as I mentioned, although we make up 60% of the cancer deaths, we only make up 10% of the research. Before we characterized our patients in 2015, there were only about 2% of data in the global database focusing on Asian breast cancer patients. However, together with Korea, China, and Singapore, and with our study, now the databases for breast cancer genomes make up about 20% of Asian patients. So what can we do with these Asian genomes? How do we now move forward, and how can we find the differences between these genomes in order to come up with new therapies or new biomarkers for existing therapies? So while we were characterizing our cancer genomes, we found that a particular gene had a common deletion polymorphism in the ApoBAC3B gene, or what we call the A3B. And because of this deletion polymorphism, this polymorphism creates a chimeric enzyme, and this enzyme introduces more mutations into the cancer cells. And this polymorphism is very unique because we find that to be 60% of Asians carry this polymorphism, and only 15 and 2% of Africans carry this polymorphism. And because A3B carriers are known to have hypermutated cancers because they are more active, they introduce more mutations, they have higher neoantigen load, meaning more mutations, more proteins that are new, that the immune system has not seen before. And because of this, patients who have higher immune gene expression and also an enriched immune activation. And we hypothesize that because of this, our patients should have an increased response to immunotherapy. And we proposed a trial that was funded by charity in metastatic breast cancer HER2 negative patients. And these patients were selected for the A3B deletion and then treated with anti-PD-1 therapy. The trial has just concluded, and we're really happy to report that with this biomarker, we've increased the response rate, or objective response rate, 2 to 4 times compared to that reported in unselected patients in the literature. And our median overall survival with the selection has also increased from 9 months to 15.2 months. So this is an example where, by understanding the genomes of our patients, we are now able to propose things that are unique to our patients. I'm going to now move on to talk about head and neck cancers, or oral cancer, or mouth cancer. So 74% of oral cancer deaths occur in Asia. So 3 out of 4 people who die globally of oral cancer are going to be in the Asian population. And looking at the genomes of this particular cancer, there's actually very few actionable mutations. So no EGFR mutation, no BRAF mutation, PIK3CA mutation also low, HRAS mutation also low. So what are we going to do with this cancer? I'm going to talk about how we look at gene expression in order to identify antigens that could be targeted by cancer vaccines. But before I do that, again, I want to point out the mutational profile, although quite similar between Caucasians and Asians, have some peculiarities in Asians. For example, the FAT gene and the caspase-8 gene mutations are more commonly found in the Asian population. What is the implication of this? Caspase-8 mutation is an apoptotic gene, and having this mutation means that the cell no longer dies based on the apoptotic pathway. So how does the cell die? It uses other forms of cell death that are more immunogenic, and therefore patients with caspase-8 mutation may be more amenable to immunotherapy as well. So before I go on to talk about cancer vaccines, I just want to talk a little bit about targeting the tumor immune microenvironment. And immunotherapy is now already approved for more than 25 kinds of solid tumors. And this is important because you can see that amongst the tumor cells, or the tumor microenvironment, is not only consisting of tumor cells, but many different kinds of immune cells, such as the T cells and other forms of immune cells that should be able to keep the tumor at bay. And the most forward immunotherapy in the clinic right now are checkpoint inhibitors, such as anti-PD-1 or anti-CTLA-4. And over the years, you can see that the number of different kinds of solid tumors amenable to checkpoint inhibitors are increasing tremendously. So about 43% of the patients are actually amenable to immunotherapy. However, if you look at the real world data, only about 13% respond to these forms of checkpoint inhibitor. So what we want to do is, how do you increase this number of people responding to meet the people who are supposed to respond? One way of doing this is to activate the immune system via a cancer vaccine. So this work is led by Dr. Lim Hui Peng, and what we try to do is to collect oral cancers from patients, characterize their genome, and find important genes that drive the tumor. And what we wanted to do is to develop vaccines and test this in animals and cell line models. So what we found was 2 very special cancer-associated antigens called MHD4B and FJX, and we found these to be highly expressed in the large number of head and neck cancer patients. And you can see here, we also confirm the high expression of these antigens, both MHD and FJX, using the genomic data that I talked about from TCGA. And you can see whether or not the patient is a HPV positive or HPV negative head and neck cancer patients. These antigens are highly expressed compared to the normal. So what we did was we synthesized several generations of cancer vaccine. First, a peptide vaccine. Secondly, now we are working with a DNA vaccine. And vaccinating animals with tumors expressing these antigens managed to increase the tumor infiltrating lymphocytes, meaning the T cells now recognize the tumor as a foreign body, and these arrows basically show that the tumor cells have already infiltrated compared to unvaccinated animals. And looking at the tumor size, this is the control, unvaccinated animal. This is our DNA vaccine. This is the approved anti-PD-1. In combination, our vaccine and anti-PD-1 almost reduces the tumor growth almost by 97%. So currently, single agent or monotherapy of immunotherapy only benefits about 16% of patients. And what we hope to do with anti-PD-1 and our vaccine is to increase this response by far. How about other cancers? Will other cancers also be amenable to this cancer vaccine? The answer is yes. So we look across many different kinds of common tumors, and in particular, we are interested in lung cancer as well. So what we have done now, we have identified the antigens. We have done the functional studies showing that actually these antigens, when overexpressed, causes cancer cells to proliferate faster, migrate and invade into different organs. We have created several generations of cancer vaccines, demonstrated their immunogenicity, efficacy, and also that they are safe in animal models. And we're currently licensing these patents to a DNA manufacturer. And if all goes well, we will be launching the phase 1 first-in-man trial called the Phoenix trial, together with Professor Ottensmeier and Professor Salveira in the University of Liverpool. So before I finish my talk, I just want to focus a little bit about germline sequencing, because I think this is something that will be of interest to the audience. And again, germline sequencing will help us identify and stratify women for better screening. So previously, about 11 years now, we have this very sensational news about Angelina Jolie doing a prophylactic mastectomy. And many people then begin to know about what is the BRCA1 and BRCA2 gene. But at the moment, we know that breast cancer is way beyond BRCA1 and BRCA2, and there are many other predisposition genes. And because we have so much advancements in molecular genetics and therapeutics, this has really greatly influenced the practice of genetic counseling and testing, whereby the cost of genetic testing is markedly reduced, making the test more accessible. There are many other predisposition genes, making this a multi-gene testing. And it goes beyond screening, because if you detect mutations, some of these even influence therapy. And as Professor Leong put it, whether we like it or not, we have to be ready, because our patients demand it. So this is the clinical practice guidelines in Malaysia for the management of breast cancer. And I just want to illustrate to you how fast the field is moving. And in 2019, this document is dated in 2019, and this is a table extracted from this CPG. And this table was derived from a publication in 2015, talking about other predisposition genes that we may need to think about when we are doing genetic testing. And in the CPG guideline, it says BART1, RAD51C, and RAD51D has been shown to be associated with increased breast cancer risk, but the risk remains inaccurate because this is a rare variant. But in 2021, we and others published an important paper, and this paper is very well populated, not only by Caucasian genomes or genetics, but also with Asian genetics. And I just want to point out the Malaysian team led by Prof. Teo, and these are the team, and of course you will recognize Sok Yi, who spoke in the previous session. And in this critical paper, 8 genes were found to confer increased breast cancer risk. 3 of these, no surprise, BRCA1 and 2, and PALB2 are high-risk genes, and 5 other genes had moderate risk. And now this includes RAD51C and RAD51D, and also BARD1. and there are risk management implications for all of these genes. So this is the importance of doing research and making sure that our patients are included in seminal studies, and these genes now should really be considered by the CPG group to be included in the multi-gene panel testing for breast cancer. So in summary, all these characterizing of cancer genomes are really expensive, but they are important because they lead to precision medicine, not only for the treatment of cancers, but also for stratifying risk. And we should leverage on population-specific genomic information where we can develop therapeutic biomarkers, identify new therapeutic targets, develop new therapies, and also influencing clinical practice guidelines in our population. I just want to acknowledge our patients who participate in our cancer research and their caregivers, where in their darkest hour, they are still brave to take part in research. Of course, our donors and our collaborators, and the team at Cancer Research Malaysia. And before I end, I just want to give a shout out to some of the charity events that are coming up. Terry Fox Run to the Malaysians next Saturday. And Bald and Beautiful, if you're brave to go bald, please join me on the 13th of October. Okay, thank you very much for your kind attention.
Thank you, Dr. Chan. That was an outstanding talk demonstrating the power of genetic testing impacting therapy directly. So we'll move on to our next and final speaker, Dr. Pacino. As you have heard, he's the Vice Chair of the Department of Molecular Genetics at Baylor College of Medicine, and he's going to talk to us about treatments for achondroplasia.
Thank you, Dr. Lee.